let's go then. Thank you so much to all of you for attending this, for being here today. Welcome to this session on biological dispersal. And in this session, we will learn from several panelists about processes and patterns of species distribution and ecological and genetic connectivity of populations in Antarctica, both terrestrial and marine. And I am Chiara Papetti. I am a scientist at the University of Padova, which is located in Italy. And I will now pass you over to my co-chair, Jamie. And I hope you all enjoy the session we have put together today. Thank you very much.
Yeah, I'd also like to just welcome you all. I'm Jamie Maxwell. I am in the National University of Ireland, Galway, and also associated with British Antarctic Survey as well. So yeah, while I'm here, I'd like to also encourage people to go to the poster hall and see any posters which are associated with this session as well, because there's a few in there as well. So we're going to kick off with a pre-recorded talk. Unfortunately, Alice could not be with us. So this is Alice Goodsey, and her talk is on DNA barcodes assessment of Ophlithplinthus based on mitochondrial CO genes, and reveal multiple cryptic species within the Southern Ocean. So I think if we can play that talk, that would be great. Is the IT department, do you have that up and ready to go? The first talk by Alice.
Not yet, but in case we have it.
You do have it, yeah.
Yeah. If it doesn't start, okay, let me see. I can share my screen and let it play.
You have it, do you?
Yeah, to introduce the next speaker, who is Jamie Maxwell from the National University of Ireland, and he will tell us about sea spiders.
Okay, hello everyone. Oh, too far. So, as you know, my name's Jamie, and I'm here to talk about the role of water mass properties in structuring Antarctic benthic communities, a study using pycnogonids. So, the aims of this project were to test whether we could using physical parameters to define faunal groupings for multivariate analysis, we'll be able to capture finer scale patterns lost when more traditional equal area or locations are used. And then to see if any of the assemblages that we got could be associated with known Antarctic water masses. So firstly, to give you an idea of what I'm talking about losing finer scale patterns, here's an example from the tip of the Antarctic Peninsula where we're using equal area hexagons. And as you can see, there's a large variation between the sampling effort and also in the physical parameters. So if you're using this analysis, any patterns associated with any of these parameters is going to be lost due to smoothing. So we decided that we were going to use temperature and salinity values as a proxy for density and see if we could capture any fine scale patterns across large areas. So before I go into methods, I just want to introduce sea spiders for those who don't know, or pycnogonids for those of you who don't know. So they're a class of arthropods, and they are Silicerata. They're a sister group to all Eucelicerata, which includes land spiders, scorpions, and horseshoe crabs. They're found globally, they're found in every ocean, and there's over 1,300 species, which are in 87 genera. And there's 10 families. Now, the reason the Southern Ocean is really good for studying sea spiders and why it was good for this analysis is that it's the only place where all 10 families are known to occur together. It's also extremely species rich, with 20% of all species being found in the Southern Ocean. Another reason why they were good for this analysis is that there's actually a large occurrence database stretching back nearly 200 years. So we had enough data for us to work with. So what did we do? Well, so first we extracted all our occurrence data, mainly, well, all from GBIF and OBIS. There was 1,227 stations, including 197 new stations records, which were the result of me identifying 5,500 sea spiders over lockdown from my kitchen table. And that was the start of my PhD in the first year, which was interesting. So in the end, we had 173 species within our data set. The purple dots there are the ones that I have added. So then we got the bottom temperatures and salinity values for every station, and we ran cluster analysis on that, which produced 18 groups. And after that, we used these groups and we put in the faunal data into these groups and reran hierarchical cluster analysis using a group average linkage to see what we got. So what did we get? Well, it produced 8 significant assemblages. We are only interested in the first 6, which are the colored ones on the left-hand side. The 2 on the right-hand side either had very low species numbers or very low sampling efforts, which we couldn't be certain that they weren't within one of the other groups. So we're just going to discuss the first 6. Below each branch, you can see the depth ranges, so quite wide depth ranges, but the black boxes represent the middle 2 quartiles, with the median depth being the white line within it. So we had assemblage 1, which is the blue one, which is roughly 50% similarity with the rest, and that's kind of the out group to everything. And then there was a further split with the other 5, with assemblages 2 to 5 being below a medium depth of 250 meters, and then assemblage 6, which is the yellow one, being above 250 meters. So there seemed to be some sort of depth related split there. But could we associate any of these assemblages with water masses? So in order to do this, we took the higher and lower values of both temperature and salinity to create the temperature salinity space, and then we mapped where these conditions occur, which is what you can see on the right hand side. So assemblage 1, it was the coldest of all, as you can see down there, and its values were closest to ice shelf water. There's also likely an influence of high salinity shelf water as well. There were 45 species within this group, of which 8% were unique to this group and not found in any other group. The second group was mostly confined to the Weddell Sea, with a bit of an overspill into the Bransfield Strait up there. And these conditions are most similar to Weddell Sea bottom water. This had the highest number of species of any assemblages, with 111 species in total, of which 14% of them were unique. Next, we have assemblage 3, which you can see circled there. The conditions where this is found is very small compared to what we've seen so far, but mostly confined to around the South Orkneys and also down in the eastern Weddell there. These are most likely areas of local mixing, so locally modified waters, where they are different water masses are mixing, and we're not sure. This has 77 species, of which 14% of these were unique. One of the really interesting things about this assemblage, although it was a small area, is that it was particularly rich for the genus, for species within the genus Colosendis. There were 29 species within the whole data set, of which 19 were found within this assemblage. Or to put it another way, 24% of all species within this assemblage were for Colosendis. So although small, these water conditions seem to be really important for this species, or sorry, for these species, for this genre. Next, we have assemblage 4, which is most like Weddell Sea deep water. It's the first one to properly kind of escape the Weddell Seas. This assemblage had the second highest number of species at 105 species, but it by far had the highest number of unique species. So 21% of the species within this were only found within this group. It also had the highest number of genre, with 20 of the 23 within the data set were found within this water group, within these conditions. Assemblage 5 was pretty easy to identify. Well, we're pretty certain it's circumpolar deep water being forced up onto the shelf there on the Western Antarctic Peninsula. This had 50 species within it, of which 8% were unique. Both assemblage 5 and 3 had the lowest number of genre present, with only 13 of the 23 present in both groups, in both assemblages. And finally, we have assemblage 6, which is the most varied in terms of conditions, as you can see there, with, particularly in terms of salinity, but also quite a wide temperature range as well. This by far had the largest sampling effort of any of the assemblages, with over 400 stations, 150 more than the next station. But there was 93 species, of which only 9% were unique. As you can see, the areas where these conditions occur are mainly on the Western Antarctic Peninsula and coastal. And because they, for the most part, are fresher, we've called these dynamic coastal waters. So what does this all mean? Well, you can see from the species accumulation curves on the left there that the assemblages more associated with the Weddell Sea have a rich Antarctic diversity. So they have a higher species richness than the ones associated with the warmer Western Antarctic Peninsula. And as you probably all know, it's expected to get fresher and warmer, but there's under climate change. So from the right-hand side there, you see I've kind of grayed out a 1 degree increase in temperature and freshened it slightly as well. So you can see a large proportion of the assemblages may be under threat. So we may see a reduction of sea spiders diversity in the future if they cannot adapt to change. So yeah, thank you. I think overall we're pretty happy with the results. It seems to show that you can use these parameters to get more finer scale patterns across a much larger area, the kind of patterns that you would get in smaller in studies. I'd like to thank anyone involved with the collection of the specimens, Dr. Claudia Orango and Ana Salas Membres for helping me with my IDE. That's my Twitter there if anyone has any questions afterwards. But if anyone has any questions right now, I will be happy to answer them.
Thank you, Jamie. There are no questions in the Q&A. I have actually a quick question. How difficult it is it to identify these species in the field based on morphology? Like in the field or in the field, the large ones like Colocynthus are the easiest because they're big. So like any big taxa. When you're actually in the field doing the smaller ones, I'd say it's about 50-50. Some it'll come down to, nearly are there spikes on its abdomen or not, which you need a microscope to see. And then some you can look at and immediately go, yeah, that's that species. Okay. I came into this not knowing anything, and I feel now that I have a good grasp of it. So it's all right. Okay, thank you. So yeah, just to reiterate now, if anyone has any questions, just type them into the Q&A at the bottom, and we will read them out. So that's how you ask questions. I think we forgot to kind of mention that at the beginning. So next we have a talk from Claudia Gonzalez Weaver, who's going to talk about how many species were hidden in broadly distributed periwinkle, La Villa Alta Ornea Calaginosa. I've murdered that. So yeah, please welcome. Go for it.
Good morning to everyone, and greetings from Chile. Many thanks to the committee of this symposium by letting me present here. So I will talk about the group of small marine gastropods, Levi-Litorina. And the title of the presentation is How Many Species, excuse me, how many species were hidden within the broadly distributed periwinkle Levi-Litorina caliginosa. Okay, as an introduction, the genus Levi-Litorina includes very small marine gastropods, and it has 21 nominal species which are currently distributed in the southern hemisphere, and particularly in the Southern Ocean. Levi-Litorina species are very abundant in rocky intertidal and subtidal ecosystems. However, some species live closely associated to macroalgae. Okay, the genus Levi-Litorina is characterized by the absence of a free-living larval stage. All the species of Levi-Litorina have benthic protected development. This means that the egg masses, the female put egg masses from where small juveniles are directly born. Okay. Accordingly, most of Levi-Litorina species are restricted to particular provinces or particular areas of the Southern Ocean and the Southern Hemisphere, with the only exception, Levi-Litorina caliginosa. This species is one of the marine invertebrates with the broadest distribution in the Southern Ocean. Levi-Litorina caliginosa includes populations in southern South America, the Falkland Malvinas Island, the Antarctic Peninsula, South Georgia, Prince Edward and Marion Island, Crozet, Kerguelen, and also Macquarie Island. So what we do in this study is we analyze biogeographical patterns in Levi-Litorina caliginosa through molecular analysis. For this, we include populations of Levi-Litorina caliginosa from southern South America, the Antarctic Peninsula, and sub-Antarctic islands, such as South Georgia, Marion, Crozet, and Kerguelen. Through this, we aim to evaluate the taxonomic status of Levi-Litorina caliginosa across different provinces of the Southern Ocean. We want to understand evolutionary relationships in Levi-Litorina and biogeographic processes associated to the origin and diversification of this group. We also want to infer the potential role of vicariance or long-distance dispersal in the evolution of the genus. And through this, we aim to improve our understanding about the evolution of Levi-Litorina across different Antarctic and subantarctic provinces of the Southern Ocean. Okay. Multi-locus phylogenetic reconstruction based on mitochondrial and nuclear genes reveal the presence of at least 7 lineages within the nominal species Levi-Litorina caliginosa. The levels of mitochondrial divergence among these Levi-Litorina lineages range between 3 and 20%. Following this, we performed species delimitation analysis using 3 different methods, and they all corroborated the phylogenetic reconstruction and recognized them as different evolutionary units or species. Most of the diversity recorded within Levi-Litorina was found in the southern tip of South America with 6 different species. So following this, before this study, Levi-Litorina was a poorly represented group in South America with only 1 species in continental South America, Levi-Litorina caliginosa. After this study, we can assure that we have at least 6 different species in the group in South America. Okay, geometric morphometric analysis recognize also significant difference among these caliginosa lineages or species, and accordingly, Levi-Litorina doesn't fit to a classical model of cryptic speciation. Molecular evolution in the group was accompanied by morphological diversification. If you want to know more details about this, please visit Sebastian Rosenfeld's presentation tomorrow at the Genomic Insight into Past and Present Antarctic Biodiversity Symposium. Then, divergence time estimation in Levi-Litorina suggests that the separation between Labilacunaria, the sister genus of Levillitorina, and Levillitorina occurred around 57 million years ago. After that, the most recent common ancestor of de-analyzed Levillitorina lineages occurred approximately 38 million years ago. Following this, Levillitorina diversified in Antarctic Peninsula and southern South America between 20 and 10 million years ago. And finally, Levillitorina also diversified in southern South America between 7 and 2.5 million years ago. We also detected evidence of recent long-distance dispersal between Antarctic Peninsula and subantarctic island populations of Levillitorina. We found one lineage of Levillitorina who is broadly distributed across Antarctic areas and subantarctic areas. So this represents the first evidence of a trans-Antarctic polar front connectivity in a shallow marine benthic invertebrate. So following all these results, we can now say that the endemic species found in the Falkland Malvinas Island correspond to Levillitorina latior. Then we also find that the species that is currently present in Antarctic Peninsula, South Georgia, Marion, Crozet, and Kerguelen Island correspond to Levillitorina venusta. Following this, the species found in the Beagle Channel, South Georgia, Cape Horn, and the Falkland Malvinas Island represent Levillitorina caliginosa sensu stricto. And then we have 4 species, 3 in the Strait of Magellan, and 1 in Hornos Island, located in Cape Horn, that correspond to new species, new to science at least. And they require their respective description. We are already working in this study. And as general conclusions, the nominal species Levillitorina caliginosa includes at least 7 evolutionary lineages of species. We detected marked morphological differences among the analyzed Levillitorina lineages. In terms of biogeography, the Antarctic polar front represents an historical Vicarian barrier between Antarctic and South American lineages of Levillitorina. The origin and diversification of the analyzed Levillitorina lineages in these provinces are congruent with the separation of continental land masses. Then the Antarctic polar front doesn't seem to represent an effective barrier between Antarctica and subantarctic islands, populations of Levillitorina venusta. So we have evidence of long-distance dispersal in this marine gastropod. And South Georgia represents a very interesting biogeographic area for Levillitorina, because in this island, we can find together lineages from southern South America and from Antarctic Peninsula together. Still to do, we have to include in the analysis more species from the Antarctic Peninsula, Levillitorina wandelensis, and from South Georgia, Levillitorina granum and Levillitorina pygmea, in preparation. To include also Levillitorina species from other provinces such as Australia, New Zealand, and of course, Heard Island and Macquarie Island. And we still require to perform contemporary connectivity analysis using SNPs, for instance, within Levillitorina venusta populations from the Antarctic Peninsula and subantarctic islands, also in preparation. And all the results I have presented here has been recently published in Journal of Biogeography. This manuscript was recently published. In fact, this is the August number of Journal of Biogeography where our study was published. And of course, the acknowledgement, and that's it. Thank you.
Thanks very much. We have one question in here. What about the South Shetland Islands? Did you include anything from there?
Excuse me, I have a problem with my... From the South Shetland Island, yes, of course. We already analyzed more than I think that we have almost like 200 specimens. Most of them were collected from the South Shetland Island. We have individuals from King George Island, from Robert Island, Greenwich Island, Deception Island, and also from the Antarctic Peninsula. And we have recorded in the Antarctic Peninsula 2 different lineages. One corresponds to Levelitorina venusta, this broadly distributed lineage. But we also have in Antarctica another lineage that includes 3 different nominal species. So on the one hand, we started with 1 species that finally resulted in 7 different species. And on the other hand, we include 3 different from the Antarctic Peninsula: Levelitorina claviformis, Levelitorina antarctica, and Levelitorina umbilicata. And they all resulted in the same clade. So it looks like I have one more here. Here we have Levelitorina antarctica, Levelitorina claviformis, and Levelitorina umbilicata. And in terms of genetic, using mitochondrial and nuclear genes, they are all the same. So it looks like we have here, instead of 3 species, 3 nominal species, we have only 1 evolutionary lineage. But these are preliminary data. We only have like 10 specimens per species, so we require to increase the number of individuals, and of course to try to include in the analysis the type localities of these species.
Brilliant, thank you very much.
Thank you. And now we move on with the next speaker, who is Valentina Bernal Duran from the University of Chile. And she will tell us about connectivity in nototenioids, and in particular in the genus Arpagifer.
So, hi everyone, good morning. You can hear me, yes?
Yes, we can hear you.
Yes, I'm going to talk about limited population connectivity along maritime Antarctica, and dispersal barriers and genetic differentiation between South Georgia Island and the Antarctic Peninsula in the nototenioid fish Arpagifer. This is part of my PhD thesis. So I'm going to start talking about connectivity and dispersal. And we like to think in dispersal in 3 stages. First, potential dispersal, that talks about larval dispersal trajectories and transport. And we study this principle using biophysical models. Then we have realized dispersal that occurs when it is dispersal, but not necessarily successful reproduction of individuals that migrates from one population to another. And we can study this with parentage or assignment test using genetics. And at least effective dispersal that occurs when is successful dispersal and reproduction from individuals from one population to another. And this is where we are talking about effective connectivity. And we also study this with genetic analysis, for example, isolation by distance. So why maritime Antarctica? We like to call this area like this because we have South Georgia Island in the north part of this map, Sydney Island, and several populations in the West Antarctic Peninsula. And we are also talking about one evolutionary lineage that includes individuals from all this population in the West Antarctic Peninsula and also from South Georgia. We made in other investigations analysis with CO1 genes, so we didn't find differentiation between these 2 nominal species, Arpagifer antarcticus and Arpagifer georgianus. So we are, they are all part of the same group. So we are taking this group and analysis more fine scale. So we are talking about Arpagifer antarcticus, which is a species with a complex life cycle, which means that they have one phase in the benthic habitat and one phase in the pelagic environment. So these species put benthic eggs, like we can see in this photo on the top. And there's always an adult taking care of the nest. And then from this egg, it hatch a pelagic larval of a shorter pelagic larval duration compared to other notothenius fish. For Arpagifer antarcticus, it's about 3 months compared to other species that have 6 months of pelagic larval duration. And it had been described that hatch periods can vary with environmental variability. The hatch periods are not the same depending on different localities. So all of this lead us to question if there are limitations to population connectivity from different localities of Arpagifer antarcticus lives. So we are combining approaches. First, to study potential dispersal, we use a biophysical model, specific Lagrangian simulation, to simulate larval transport between 10 localities, including South Georgia, Sydney Island, and different localities in the Antarctic Peninsula. And we set the hatch period between November and February. That is the hatch period that had been described for Arpagifer antarcticus. And we followed these Lagrangian particles from 100 days. That is the pelagic larval duration for this species. Then for study effective dispersal, we use genetic approaches. We study more than 20,000 neutral SNPs obtained by GBS from 13 different localities. And we study population structure and gene flow to see contemporary connectivity. So this is the map of the different localities that we have samples and that we include in the biophysical model. For the circles, we have all the samples that we have from genetic or genomic analysis and biophysical model. There are also other localities where we also only have genetic information, but we didn't include this in the biophysical model because it's very near to other localities. And one locality that is only included in the biophysical model because we didn't have samples for genomic analysis. So this we have a total of 170 samples for the genomic part. And then some results and discussion that we have right now for the biophysical model. You can observe here the particle tracking from the West Antarctic Peninsula to Sydney Island here and South Georgia Island from November and December. And we can observe that there's no particles that travel from West Antarctic Peninsula or Sydney Island that can reach South Georgia, and the particles released in South Georgia show this trajectory to the northeast, so they also can't reach to the south. And here we have a difference between this November and December results, and then from January and February we can find some particles that travel north from the Sydney Islands to the South Georgia Islands. So it could be that some particles, maybe a small part of one, in this case representing a larval dispersal of Arpagifer antarcticus, could reach South Georgia if they hatch later in the period. We're talking about like December, January, or February. So for the population structure, we made different analyses. Here we have 3 different analyses. First, the discriminant analysis of principal components. And if you can see here, we have South Georgia individuals that are differentiated from the West Antarctic Peninsula here on the right. And in green colors, we have the north part of the Antarctic Peninsula, including the Shetland Island and some populations in the north part of the continent of the peninsula. And in orange and red colors, we have the central part and the south part of the West Antarctic Peninsula. So we have at least 3 groups in this all data. In other analysis, we also made a structure analysis that also differentiate South Georgia from the samples in the West Antarctic Peninsula. But we can also see some genetic exchange between individuals. This is why we have different colors and in the different populations here. So this analysis can also differentiate South Georgia. For the gene flow analysis, we took these 3 putative groups, like one is South Georgia, the second one is the north part of the Antarctic Peninsula, including Sydney Island, and then the central and south part of the Antarctic Peninsula, like a third group. And we saw how is the gene flow and the contemporary connectivity between these 3 clusters. So we found that between the North Antarctic Peninsula and the South Antarctic or Central Peninsula, we have some exchange of genetic information, but now with South Georgia, it's very low, the exchange of individuals. The same in this case. This analysis show also a high proportion of individuals that settle in the same population that they born. So there's no exchange of genetic information between South Georgia Island and the West Antarctic Peninsula, talking about in contemporary terms. So we found some similar patterns of that I'm talking about here for Antarctic species, Pachyphyra antarcticus. We observed some similar spatial structuration in sister species, Pachyphyra bispinis, from the Patagonia, the south part of South America. And we also found 3 clusters. This research was published recently, and we found long gene flow between these 3 clusters also. It's a very similar pattern that we observed with Pachyphyra antarcticus. Nevertheless, in this investigation, we didn't include a biophysical model as we are including now with Pachyphyra antarcticus. So as main conclusions, we found congruent results between biophysical model and genomic analysis that shows that there is some limited potential dispersal between South Georgia and West Antarctic Peninsula. This is the principal due to the oceanographic conditions in this area that impede individuals' dispersal from South Georgia to West Antarctic Peninsula, because the direction of the currents there is from northeast, not to the other direction. So this impedes that the larvae that hatch in South Georgia travel to the south, to the West Antarctic Peninsula. And some biological limitations to dispersal from the Antarctic Peninsula to South Georgia, because we have the direction of the currents to the north, but it seems that 10 days of pelagic larval duration is not enough to reach one population to another. And we found, due to all this population structure and limited gene flow, that this is why we have limited contemporary connectivity between these 2 areas, that it's South Georgia and the West Antarctic Peninsula right now. So announcements to different institutions and projects that made this investigation possible. And thanks all for your attention. And this is my emails if anyone have questions or have to contact me. Thank you very much.
Thank you very much, Valentina. Very interesting. There are no questions in a Q&A session, but I have a very quick one, just a quick one. Do you plan to look at any temporal aspect of this differentiation, to look at the age of the individuals?
We have tried to read the otoliths, but as we have most of our samples are adults, it's very complicated to access to the micro increments in the otoliths. So it has been very difficult to access to the age. But we have interest in this because we also want to know if there are different hatch periods, and we can saw this mark in the otoliths of the individuals.
Yeah, thank you very much.
Thank you.
Okay, next we have another pre-recorded talk, and this is from David Barnes, and the talk is entitled Understanding Past and Future Connectivity. Are Under Ice Shelf Samples Key? So if the IT could play that, please, that would be brilliant. Thank you.
Hi, and welcome to the talk by me, my RV, Oxford, Bass and Padua co-authors on past and future high Antarctic marine connectivity. We ask under what conditions could or has the West Antarctic ice sheet, WAIS, collapsed. Such an event would lead to more than 5 meters of sea level rise, a new seaway across the continent or seaways, and considerable likely disruption to flow, heat and oxygen around and beyond Antarctica. It would also give considerable impacts on organism dispersal on connectivity, and we think that signal might be preserved. Biology, particularly on the seabed because of lower mobility, can preserve strong past connectivity signals. Bryozoans, for example, have sessile adults and short-lived benthic larvae, and this can strongly limit dispersal. You can see the variety of form that bryozoans take in the top right-hand side. In the main plot, we can see the samples containing bryozoans around the Southern Ocean and nearby areas. We can look at the distance apart of those areas, shown on the left-hand side around Antarctica, with and without putative seaways. And we can compare similarity of bryozoan assemblages with distance apart, with or without those seaways in the models. So if we do that, the most obvious first result is an anomalous similarity of Weddell and Ross assemblages, seen in dark blue in the middle. That would imply past connectivity as suggested. The most similar other two regions around the North Antarctic Peninsula were thought to be ice-free during the LGM, the last glacial maximum. So is this a clue to past seaway connectivity? Multidisciplinary effort a year later added samples from the Amundsen Sea and looked at ice mass fragility pathways. And we've done this across a variety of disciplines there. So let's have a look at that and more in the box in detail at what possible pathways might have been taken. Well, oceanography can inform us about the strength, duration, predictability, temperature and freshening of currents that bathe the ice shelves. Geography can map the seabed elevation underneath ice. They're seen in orange, such as with LiDAR. Glaciology can show ice thickness and thinning rate, and crucially, the amount of ice that needs to be thinned for instability. And the most fragile pathways, if we do that, seemingly could have collapsed with temperatures that we know happened in the last interglacial and within 1,000 years of recent warming patterns, but that is of course accelerating all the time. And we can see those in the hard black lines in the map on the right hand side bottom. So let's plot similarity of bryozoan assemblages on the y-axis with distance apart on the x-axis. And we can see in the graph above without seaways, i.e. with the waste intact as it is now, and that below with seaways put in. So what difference does this make in terms of looking at our models there? Well, similarity of bryozoan assemblages is best predicted by proximity. The way seaways better explain modern distributions, as can be seen by the increase in R² going from the top plot to the bottom, and likewise in the ANOVA associated F, but also in the reduced probabilities. The best fitting model is that with the Amundsen seaways included, and that agrees with the glaciological pathway fragility shown by Vaughan et al. 2011. However, these analyses are compromised by the paucity of sampling at each end of the putative seaway. The problem is that these are rarely sampled, difficult to reach location, with much of the closest points underneath ice shelves, hard to access. Sampling under ice shelves is very difficult, costly and time consuming. But various drill holes, such as around the Neumeier 3 station, have shown glimpses of occasional larger animals on the seabed. But 2 recent drill holes here have actually yielded macrobiota samples, as you can see in that cup, and a lot of it. And this has allowed us to actually look in detail at the species and some of their characteristics. So here we can see the location of such drill holes on the Ekstrom ice shelf. And here we can see some of their characteristics. So a recent study showed that they were much richer or as rich as normal shelf samples, a big surprise, particularly in these bryozoans that don't move around much. Not only that, but analysis showed normal growth levels, remarkable considering they are kilometres away from in situ primary production and in perpetual darkness. They also showed persistence for long periods. Nearly 6,000 years was the oldest of the samples in that cup. Thus, drill holes through key places have a good chance of yielding strong benthic samples. And we can see pictures of the seabed around the bay there. So, multidisciplinary approaches all seem to point to the same places to tell us most about past connectivity and when this last was, thus giving a strong steer to future tipping points in a current warming world. Collecting new samples near these likely old and future seaway gateways or under ice shelves as close as possible to them should help considerably in establishing when they were last opened and are likely to be again. And with that, I would like to thank many co-authors listed here, and particularly those on the RV Voyage PS129, which allowed me to actually see these field samples for the very first time. Thank you.
So great, and once again, I recommend, or I encourage you to contact the authors of the pre-recorded videos in case you have questions for them. And now we move to the next speaker, who is Andrea Pinones from the University Austral of Chile. And Andrea, are you there? See? And Andrea will tell us about hardcore circulation. So especially about the circumpolar deep water. Thank you, Andrea.
Thanks, Chiara, for the introduction. So can you hear me?
Yes.
Yes. Okay, great. Thank you. All right. So this is a collaborative study done with different colleagues from different institutions, primarily from the US and here in Chile. Some of the institutions are based here. I ran the biophysical couplet processes lab at the Universidad Austral de Chile in southern Chile. And I also participate in different research institutions that are mentioned over here. So like Chiara said, my study region is the Antarctic Peninsula. So in this region, one of the most dominant ocean circulation features is the Antarctic circumpolar current. This current runs from west to east, and the southern boundary of the ACC aligns pretty much with the continental shelf break along the West Antarctic Peninsula. Okay, one of the important features here is that the ACC transport circumpolar deep water. This is a relatively warm, salty, nutrient-rich water mass, okay, that plays a significant role in different physical and biological processes in the continental shelf. Now, these circumpolar deep water intrusions occur along deep bathymetric depressions and marine canyons that are placed along different regions on the West Antarctic Peninsula. So it's a way of connecting the open ocean with the inner shell, and particularly with the inner shell. So in our study region, which is located here in the North Antarctic Peninsula around the South Shetland Island, we are also interested particularly in an embayment that is located in King George Island. This is called Maxwell Bay, that is facing south into the Bransfield Strait. So in this region, the circulation can provide environmental conditions that are favorable for the formation of biological hotspots. What are these biological hotspots? Well, these are, like I'm showing you here in the right plot, all the squares in red, denote or show locations of this area, of these biological hotspots that are regions of persistent richness of marine species. Some of the marine species are named or are showed here. So as we can see, the ocean circulation has the potential for transporting and playing a significant role in the connectivity of organisms in this region. And so this is what we wanted to accomplish in this study. So the objectives were to first describe the intrusions of circumpolar deep water from the open ocean, from the Bellingshausen Sea into the Bransfield Strait, and potentially into the region, the embayment that we are interested, which is Maxwell Bay. And also we were interested in determining and perhaps quantify pathways of connectivity around the South Shetland Island. How did we do this? So the approach we did was to implement different experiments, simulation, basically Lagrangian particle experiments. We also did experiments using tracers, and also we analyzed historical temperature and salinity observations done in this region. I'm gonna very quickly provide you some of the methods that we use. So like I said before, we did ocean modeling and analyzed historical observations. The model we used was the Regional Ocean Modeling System implemented for the West Antarctic Peninsula. This model has a high resolution of 1.5 kilometers in the horizontal. Is composed of different sigma layers at depth and also includes tide. If you wanna look at more detailed information, I encourage you to look at this study by Kate Hudson, recently or relatively recently published. This ocean model also has not only ocean circulation, it also includes sea ice dynamics and also the effect of ice shelves, okay? And like I said, we did different experiments using passive tracers. Basically, we use a dye to represent the advection of circumpolar deep water. And also we did Lagrangian particle experiments that were released outside the embayment that I was mentioning you about. And the observations we analyzed thanks to the collaboration with researchers from NOAA. We obtained the Antarctic marine, from the Antarctic Marine Living Resources. We use historical CTD observations from summer, from winter. We also analyze temperature and salinity data obtained using using tags that were deployed on weather seals. And finally, we also analyze or use CTD observations from summer cruise inside the Bay of Maxwell. So the following animation I want to show you here is the simulation we did with the passive tracer, okay? So like I said before, this passive tracer is meant to represent the advection of circumpolar deep water along the study region. And so some of the interesting things here is that this intrusion has a very clear pathway along the study region. These pathways start from here. I'm trying to show you the initial point of intrusion, which is between Smith and Snow. Then it follows the bathymetric contours, goes around Deception Island, south of Deception Island, and follows the pathway to the east along the shelf break of the South Shetland Island. There are also the other interesting feature here is that here the point, the dot, the blue dot, shows you where the simulation is running, what year the simulation is running. And the scale here is basically the vertical mean of the dye concentration that it was quantified in between these smaller squares. So in order to show you along the pathway and along the advection of this CDW, how this gets eroded, right? So basically what I mean here is it's not the same amount of CDW that you find in the open ocean here, where the gray line is, where the gray square is, compared with what is basically available in front of Maxwell Bay. In order to sort of compare and validate the simulations we did, we analyzed the historical CTD observations along the pathway of intrusion, and we took different areas basically along the pathway that I was already showing you. And so the colors on the CTD profiles are coherent with the pathway from the open ocean into the South Shetland Islands. It's what we can see here. And basically, I want to drag your attention into the salinity temperature diagrams. Is that right over here we have as the temperature and salinity characteristics of the circumpolar deep water. So circumpolar deep water is available and is present, and we can see it in the observations, basically with a very strong signal on the point of intrusion, right? But that signature gets eroded, right? It gets mixed with other water masses, and therefore what you observe as you can go into the embayments over here is that you observe a modified version of this circumpolar deep water. Now, but what happens inside Maxwell Bay, and what is the potential of this modified circumpolar deep water to get intruded or get inside Maxwell Bay? And so here we analyze all the temperature and salinity that we were able to analyze from different years, and we divided into inside Maxwell Bay and outside Maxwell Bay. And here we observe also the temperature and salinity characteristic for this modified version of the circumpolar deep water. And what we observe is that outside the bay, there are more observations that are closer to this modified version of the CDW. But as we go inside the bay, basically the TS diagram doesn't show a clear signature of this modified circumpolar deep water inside the bay. And this is consistent in winter, spring, and summer. In summer, the modified outside is a bit stronger, but not as much. Okay, and so, but what happened during time? So in between, here inside the bay, this is a time series of salinity and temperature inside the bay between August and November. It's very difficult, and basically we didn't observe modified circumpolar deep water, even though we did it, or we analyzed a time series where a stronger intrusion was observed during the simulation. So I encourage you to also look at the poster by Paula Amador-Bellis. She also is gonna show some of the physical forces that are perhaps influencing these patterns locally. And finally, the final figure, before I go over time, is to show you what happens from Maxwell Bay into other regions, how whatever is happening in that region and the coastal embayments of the bays south of Kingeorg-Tyler are connected to other regions around the South Shetland. And this is shown by the particle transport. So basically the export from Maxwell Bay into other coastal regions are taking about days and up to 1 month and get connection or connectivity between this embayment and around the South Shetland into the northern part. In summary, pathway of intrusions of the CDW from the Bellinghausen Sea onto Maxwell Bay are defined along the shelf break south of the South Shetland Island. We also look at and show that observations of CDW along this pathway are consistent with the pathway shown by the simulations. The CDW gets eroded along this pathway, so the signature of this is basically a modified version of CDW, and is relatively weak at Maxwell Bay, so the potential influence into Maxwell Bay is low. And the temporal scale of connectivity from outside the bay into other coastal embayments of the North-South Shetland Island ranges from days to the scale of 1 month. This has implications for the connectivity of planktonic organisms locally, and also can contribute to understanding of possible connectivity mechanism from the bay to other coastal environments. And these are my acknowledgement to our National Agency of Development and Research, and and also INACH, who provides great support for all our Antarctic research studies. And there is my contact information if you have any other further questions. Thank you.
Thank you. I think there is no time for a question now, and I pass you over to Jamie.
Okay, move swiftly along. Next, we have a talk from Claudia Maturana, and it's entitled Assessing Habitat Suitability for Bocella Popeye Across Antarctica and the NSA ecoregion. So go ahead there.
Okay, here. Do you see my screen?
Yeah, we can see it.
Okay, perfect. I guess I'm going to start video. Okay. Yeah, thank you very much for attending this presentation. I'm very glad to be in this session. I'm surrounded by a very good friends. And yes, we are going to talk about habitat suitability and contemporary spatial structure of biodiversity, which is which has been strongly influenced by natural, climatic, and tectonic changes that have occurred over long time scale. So, however, Antarctica now, its surrounding islands are showing alarming response to the effect of human climate change. Today, we are facing one of the greatest threats to Antarctic biodiversity persistence. So Lee and collaborators in 2017 evidenced that Antarctica is melting at different rates and different areas, showing the Antarctic Peninsula being the most affected area. So as a consequence of this, we expected to see an increase of an ice-free area. So this is particularly important because these areas host almost all terrestrial and freshwater biodiversity. So we need to increase our understanding of how biodiversity change is driven by climate change over space and time, and we need to take this task urgent. So the niche ecological models of suitability are expected to hold some predictive power across space and time. But before using this tool, we need to consider two important challenges. First of all, we need to build a niche model that is mainly based on environmental variables which can be transferred across time to predict future suitability areas for the species. And second, we need to use dispersal and demographic knowledge considering the area that is suitable for species, not only regarding its occurrence and dispersal, but also the possibility to persist and establish. So in this study, we are going to be more focused on the first challenge. So we decided to study a key species for the freshwater ecosystem, Bocchella popei, which is a copepod, and more importantly, it is the only macroscopic terrestrial invertebrate species that is known to be today distributed across the three main biogeographic regions in Antarctica and in the Magallanes subantarctic ecoregion. This species has a very limited dispersal capacity, probably mediated only by passive vectors such as birds and probably wind. This is a very, very short and simple way to describe and illustrate the niche ecological model methods. We first use the occurrence of the species dataset using GPS point with or plus climatic variables. We choose in this case 7 bioclimatic variables downloaded for WorldClim platform, and those and that data goes to the Maxwell algorithm, and at the end we use this data to project in space for this part of the study to obtain the different areas that is more suitable for Bocchella to occur. So for the occurrence dataset, it is important to note that, or to say that we use a previously published data paper where we include the occurrence record for the entire genus in the Southern Hemisphere using 3 different sources. We use previous papers, GBIF records, and our own sampling effort. In this sense, GBIF facility can give you dataset which is very easily to manage and it's very helpful for this sort of a study, and it's free and it's open. So this is a very good platform to contribute. Well, this is the map that gathers the occurrence of Bocchella popei in South America and maritime Antarctic. The northern limit of its distribution is around 30 south degree here, and the southest point of occurrence is in around the 7th south degree. Here is the habitat suitability, the projection of this niche ecological model in space. The high and very high probability, which represent the 6.3% of the study area, is concentrated in 3 main locations in South Georgia, the Falkland Malvinas Island, and the southern part of South America, mainly in the Strait of Magellan and Beagle Channel and Tierra del Fuego. Here are the results for the maritime Antarctic projection. The high and very high probability of habitat suitability is concentrated in 1.6% of the study area, which is focused on the South Orkney Island and the South Shetlands in the east part of Antarctic Peninsula, close to the or surrounded the James Ross Island. So this data, or this projection in space, now can be transferred in time. So what are the possible changes that we are going to see in the landscape in this part of the Antarctica? Well, Lee and collaborators also show that under the strongest forcing scenario, the ice-free area could expand close to the 25% by the end of the century. So we are going to see a South Orkney Island become completely ice-free, and the north part of the peninsula could contain the largest amount of ice-free area. So Antarctic Peninsula, where this magnitude of increase in ice-free area could drastically change the availability of habitat and connectivity of biodiversity. So what is going to happen with Bokella Popeye then? So this is Antarctic Peninsula under 3 different IPCC scenarios. And you can see that under 2 of them, the more conservative and more dramatic scenario, will have increase of an ice-free area in the south part of the peninsula. So probably we will have to look for the current low and medium probability of habitat suitability, because in the future, probably Bokella Popeye will colonize this area, and this will increase the habitat suitability. So this can, or hopefully this data arise an important question. Will Bokella Popeye be a winner species, as some scientists have described for different species effect for the climate change? So another and very nice paper just recently become published from Lee and her team have interesting discussion about how it will impact this increase of ice-free area in terrestrial and freshwater ecosystem. Will Bokella Popeye colonize and establish across the Antarctic Peninsula? It's not necessary an increasing opportunity to colonize. We have to remember that not because the model say that is going to be suitability to habitat, it means that the species is going to effectively establish and persist. So that's why the second aim or challenge is become very important, is to unite the information, or as a complement information, we need to know the dispersal capacity, and we have to consider the genetic approach, for example, to establish a new lineage or genetic lineage. So that's it. Thanks for your attendance and all the people who contribute in this study. Thank you.
Thank you, Claudia. Well, again, please post your questions in the Q&A session, and we move on to the next speaker. And the next speaker has provided a pre-recorded video, but she's also in person. So if you have questions for the next speaker, just type them in the Q&A session as well. And so the next speaker is Catherine Short, by the British Antarctic Survey and University of Bristol, and she will tell us about the hardy tardigrades, the genus Mesobiotus. So I kindly ask the IT team to share the video recording.
Hi, I'm Dr. Catherine Short, and I'm here today to talk about some of the work I did in my PhD with tardigrades and Antarctica. Today I'm going to talk about the genus Mesobiotus and its biogeographic history within Antarctica. So why is it interesting to study the biogeographic history of Antarctica? As you can see, Antarctica has been isolated from its nearest neighbours for about 30 to 35 million years, with most of these years being covered in ice. So what did this do to the life in Antarctica? Well, it was previously believed that when it got covered in these big giant ice sheets, that all life was extinguished on the continent, and that when they retreated and melted, that animals were able to kind of colonize from outside back in. But actually multiple recent studies on multiple kind of groups of terrestrial invertebrates have shown that this is generally not the case. That most animals, like springtails or mites, have actually persisted throughout these glacial retreats and expansions and have managed to kind of survive throughout them. But as we can see, that one animal is missing from here, the tardigrade. So what is a tardigrade? They're microscopic ectodicyzoans, which means they shed their outer skin. They're extremophiles, and they can be found in multiple extreme environments, from the polar regions to underwater sea vents to tropical rainforests. They're globally common, they're found throughout the world, and they're an ancient lineage placed somewhere between nematodes and panarthropods, but this kind of changes depending on what study and model that you look at. So what about Antarctic tardigrades? There's currently about 65 plus species known from Antarctica, with more being described all the time. They're widespread and diverse throughout the continent and play an important role in the terrestrial fauna. Food and ecological webs in Antarctica are very small, and so therefore they have quite a big impact on these. Despite this, they're actually very poorly studied, and resolving the phylogenetic relationships and their colonization history in Antarctica is important for kind of understanding tardigrade evolution as a whole, evolution within Antarctica, and future biodiversity changes in a warming world. So what is Mesobiotis, the animal we're going to be looking at today? Here's a picture, one that I actually took. And the genus Mesobiotis used to be part of a much larger genus called Macrobiotis, and kind of got separated about 5 or so years ago, and is further divided generally into two groups called Harnsworthy and Thursiger. The Harnsworthy and Thursiger groups are generally described using subtle morphological characters, which are often based on the egg sculptures. As we can see here, egg sculptures are very small and delicate, can often be damaged, and it may make it actually hard to kind of place these within either or group. Mesobiotis thursiger is noted as a globally common species group and has been noted to be places like the Arctic and Africa. However, Tchaikovsky in 2012 did show that there was some species level divergence between isolated populations in Antarctica. But there is actually no strong definition or resolution to how this group is defined, with being placed into Harnsworthy or Thursiger rather arbitrarily, and this is especially true within Antarctica. So what did we do? We took multiple moss and soil samples from around the Antarctic continent and the sub-Antarctic region to try and give a good overview of the patterns within this. From areas that we didn't collect ourselves, we did use GenBank to get the CO1 and the 18S gene regions. And then within the samples that I took, we extracted tardigrades using the flotation technique, and then each tardigrade was individually sequenced for CO1 and 18S gene regions. If you would like more information on the methods that I use for this, I have a link at the end of my talk to the paper where you can read it in more detail. So let's take a look at the relationships within Antarctica itself. So I used the CO1 region to create a haplotype network to look at the relationship between these populations. Were they managing to interact with each other? Is there a lot of crossbreeding? And as we can see, that actually these CO1 areas are very distinct. The groups are very isolated and very endemic. So how does that actually relate to the map? So if we look at this green region, which is also in the top left, we can see that it kind of corresponds to the peninsula. Interestingly, it also includes the Victoria Land group. Now, Victoria Land has actually been shown to have biogeographic links to the peninsula, within the last glacial retreat, animals being noted to be able to have moved up and down the coastline. But within this region, there is actually another specific isolated population on Alexander Island. Now, Alexander Island is believed to have a completely distinct biogeographic history to the rest of the peninsula. And actually this shows in the fact that the animals there are not related at all to the ones from the peninsula. Then we have the South Orkney region, which is where this species was originally described. The sub-Antarctic being a separate region. Then we have the continental regions of Dranning Moorland and Mac Robertson Land being discrete, distinct populations. But how many species would this actually relate to? So we ran two models, GMYC and an MPTP, both in a single and multi, to see how many species there were there. So it ranges from anything from 3 to about 10. But like the haplotype network, these correspond really nicely to the biogeographic regions of the peninsula, the South Orkneys, the Mac Robertson Land, South Georgia, and Alexander Island. And if we actually take a look at the kind of percentage differences between these populations, we can see that they're actually really huge. The Antarctic Peninsula and Dranning Moorland, for example, has a 45% difference. So this is telling us that these are very distinct, isolated populations that have been isolated for a very long time. But how do they actually relate to the rest of the Mesoboetis genus? So we took a look at CO1 and 18S. This is the Bayesian models, but I do run some more. And if we have a look at how these relate, we can see that this group here and here, these are the Antarctic species, with the non-Antarctic being placed here and here, related but strongly distinct from each other. But how long ago did this occur? So this slide here was showing something that I'm currently working on at the moment, which is a relaxed molecular clock looking at when these divisions took place. And as we can see along here, that these took place about 40 to 30 million years ago, which is when Antarctica became isolated. So this shows to us that the species of Mesoboetis found in Antarctica are strongly divergent from other non-Antarctic Mesoboetis species. Even if they have been grouped previously morphologically, there is actually no evidence for them to stay linked with each other. This means, therefore, that the Thursiger and Harnsworthy Mesoboetis groups that are widely used actually have no systematic support, and this should probably be rethought. Mesoboetis is ancient within Antarctica and has radiated since it was isolated from other continents. This was the first study of its kind into a specific tardigrade group and has actually shown similar biogeographic patterns to other terrestrial invertebrates found within Antarctica. Integrated taxonomy is urgently needed, therefore, across Antarctica to address these species-level taxonomy problems. Further sampling of Gondwanan Mesoboetis species would help to fully clarify the divergence of these groups and kind of the history of where they came from. So this would be particularly samples from Australia and South America. I'd like to thank the British Antarctic Survey and the University of Bristol for helping me through my PhD and to all of the funding that I've received. And thank you for listening. If you have any questions, you can use either of these email addresses, or I also have a link to the full paper if you would like to take a read. Thank you.
Brilliant. Thank you very much, Kevin. I don't know if you do we have any questions? There's no questions in the chat. I don't know if you're there, Catherine, but I have a question about Alexander Island, if you're there.
Yes, hello.
Hi, I'm understanding correctly. So that's within the peninsula region, right? And
yes,
do you have an explanation of why? Because it's really unique. It's not even trading close to it, is it? Do you have an explanation as to why?
Yes, so it is believed that the island of Alexander Island didn't actually come from the peninsula geology-wise. So when the kind of the continental plate of Antarctica and the peninsula kind of smashed together, that Alexander Island actually wasn't part of that and had drifted from, I believe, South America, but it drifted from a separate part and kind of come into it and is now connected by ice, but doesn't actually have the same geological history. So it hasn't come in with the peninsula. So they believe everything on there has actually evolved separately from the peninsula.
Yeah, that's really cool.
Yeah.
Thank you very much. So we're going to move on to our penultimate talk. This is Hugo Benitez and his talk on unraveling the invasive patterns of the new Antarctic fly, Trichosera maculapennis, a first morphometric approach. So take it away, Hugo. You're still muted, Hugo.
Sorry. Okay, you can listen now?
Yeah, can hear you now.
Perfect. Okay, so thank you very much. So we'll hide this. Cannot see. Here we go. Let me do that. Right. Okay, so thank you very much to the organizer to invite us to this symposium. For me, it's a pleasure to present our results from an ongoing project we are doing with Trichosera maculapennis. These results are part of a master thesis of Matías Llapis from Universidad de la República in Uruguay, and it's a collaborative work we are doing, trying to understand the pattern of adaptation of Trichosera maculapennis. That's a fly that just arrived to Antarctica a few years ago. So we are connecting the combination of different approach, and for this talk, I'm trying to talk about the morphometrics approach we're doing. So Trichosera maculapennis is a widespread fly that living in Europe that have a very widespread distribution between the Arctic, Norway, Finland, some of these places, until Spain, Portugal, Poland. So having a very physiological response to temperature, probably living in the winter, -20 in Norway, until Spain, at least 25, 30 degrees Celsius. So it's a very large adaptation to temperature. So, but they arrived in Antarctica so a few years ago, this 10 years ago. The first sightseeing was by a researcher from a Chilean station in Chile for Convenors station, but that's 2017. No, that's 2007. The first officially published was in 2013 by Odile Monteiro, Ponce Leon, Picombi, and also helped by Eva Kremiska, that was a Trichosera specialist. So for us, was very worried to know that this new fly was arriving into Antarctica. After to try to take a look at the life history of this fly, we can understand that it's very could be adapted to very cold environment. But the larvae is mostly eating subprophagous and sometimes coprophagous area. The first appearance from this Trichosera maculapennis in Antarctica was officially was in the Artigas station, Uruguay, and they try to understand what are they're expanding to different places and this very quick dispersal in King George Island. So we can find now by the last treaty meeting, consular meeting, that from invasive area that already in 2022 the Trichosera maculapennis is now inhabiting in mostly the septic areas and surrounding of the station from the mostly walls station or research station in King George. The last one was discovered in Carlini, Argentina station. We have still no evidence of the presence in Brazilian and Peruvian from the Admiralty Bay, but also this found it in Andreas Arstowski Island in Poland. So this is very worried for us that this fly is also doing, and it was found also flying in the surrounding areas. So it can be very worried for us that could be also established in natural population. So about the ongoing project that also published material that they have Trichosera already in Antarctica, there's a work from Marta Potoka from Polish station. They do the description of Trichosera maculipennis in Antarctica with all the details of life history, describing the larvae that was found, also pupa, and also doing some molecular work using some simple markers like a CO1 marker to try to see relationship between European species with the Antarctic one, so the invasive one, to try to see what was the invasion pattern of adaptation, how it's also moving between different stations. Also, Luis Pertierra, from University of Pretoria, South Africa, they last year published a work looking into the tolerance that they have Trichosera maculipennis in comparison with our native Paroclus steineri in Antarctica, and see if the probabilities that could be this Trichosera could be starting to use in the environment of Paroclus, our native species. So the thing that they found that the tolerance was even higher from Trichosera maculipennis than the native from Paroclus in Antarctica. So that also works for us that if the tolerance is bigger than the native species, the way that they can disperse more between natural environment and station could be easier. Now, in the last work we are doing, we are now collaborating with Sanji Kim from the Korean Polar Research Institute. So she's also doing some molecular work that we are now collaborating using part of morphometrics. And she found using more microsatellites, she found some relationship between two lineages between the stations and natural environment in Europe. And the first lineage was found where species from the Frey station were more related with the Point Cape in Arctic, so the native species, and also connecting with Arctosky species found in Arctosky Island. But a second lineage from the molecular work was the whole or other species from the different stations were found that and related all together. Still, we need to do more work to see if they form some genetic structure could be more related with our species and try to understand how they are advances or how they adapt to live in this kind of environment. So for that, we together with a group with the Uruguayan station and from Universidad de la República from Uruguay, starting to work together to implement part of the work that Monica Remedios do in working with Pitcombe, part of a research work in the British Antarctic Survey. We try to connect to using other methodologies to understand the pattern of adaptation of Trichosera. So been working together there and trying to see if there's a morphometrics approach could us understand the pattern of adaptation of morphology to fly in different parts of the natural environment Antarctica. So we see and collect using different blue traps and different traps and collecting also by hand in the station, in the septic areas from the multiple station, also collaborating with different researchers to help us getting some samples to there. And we try to use them to work in some morphometrics approach. So we having the glue and try to take the ones that we could use for morphometrics, but also the other that we use for molecular approach. So with this, we have we use a geometric morphometric approach with the wings that we try to see if the wings are having some morphological change. So rapid morphological changes are part of normally from invasive species. So we use the approach, we try to see if the shape variation of the wings has some modification in times. So we use flies from the Uruguayan station between 3 years, 2017 to 2019, and also in a comparison with another station, was a Russian station, some Bellinghausen station. We use the wings, we quit the wings and see using 30 landmarks if the morphometrics could help us to see if there's some rapid adaptation in morphology that is correlated with the molecular approach. So what we found we used a multivariate approach to see this relationship between different years in Uruguay and another station in Russia, and we found very worried results that is also for us a really dangerous situation that the species are changing the wings in a very rapid, very fast way, where the species, the red one and green one, are from 2017 and 2018. So 2019, that are the pink wings, in this graph show that there is a very particular wings that are changing like in an arrow. And also 2019 from Russia also have a very different morphology. This is very, very interesting for us because this kind of flies could have the wings very similar with the station. There are no much differences in kilometers that they can have this very rapid morphological change. So we analyzed the size as well, using the shape, but including the size in a allometry way. And we found that the species, the individual from 2019 in comparison with 2018 and 17 the wings are getting smaller and are getting in an arrow way. In this way, this kind of morphology normally in invasive species like Drosophila or other invasive species normally are using to try to cut the wings. When the wings in an arrow, they cut the wings so they can fly easily in Antarctic environment. So they easily, we can see that these flies are trying to go outside and try to lay eggs outside of Antarctica, outside of station, and they are doing very quick. So for us, this result can combine with the molecular approach, and we're now trying to understand how they are modifying the morphology very easy, and how the molecular also following that. So the conclusion for this work, we see that the maculipennis between the few years of observation are changing very quick, the wings modifying their phenotype to adapt to lifestyle to new environment. So the correlation of genetics with the whole range of bases is a part of the future work that we're doing, connecting all as a genomic way with a morphometric way to try to understand if there are some founder effects and how they are changing. Or finally, we have just to worry and help one ask you that all researchers that working in some station, they have samples or they can sightsee the maculipennis in their bases. Let us know, because we are trying to avoid like this final picture here that maculipennis is adapting very quickly. Now we only see in King George Island, but we finally can see a more widespread population in that. So I have to thank very much to the team, to the project. We now get a grant in INACH to analyze the data, and this is a work with a lot of international collaboration, and we're still working in that. And we hope we can having more prominent results with avoiding the establishment of this flight. So thank you very much.
Thank you very much, Hugo. And it's very interesting. And this change in the shape of the wings opens a lot of questions, actually. So you're planning to do any genomics on that, or do you expect this is just phenotypic plasticity?
Well, normally when you see invasive species have a very quick phenotypic plasticity regarding the time, but here the weather and the stations have a very similar pattern of it. There's no much changes in the climate that they have already this kind of different rapid morphology. So the way to avoid that also is try to see in the genomic patterns if there's some correlation with the genomic and morphometrics, and avoid to say that it's plasticity, and try to also avoid to say that could be a rapid adaptation for flying and getting more environment. So we cross the finger that it's only plastic.
Thank you, Hugo. And we move to the last speaker of this session. We are slightly a little bit late, but all the talks were very interesting. And so we move from the invasion to the attempt of eradication of a fly. And the next speaker is Katarzyna Tolikac from the Institute of Biophysics of the Polish Academy of Science. Thank you, Katarzyna.
Thank you very much for the introduction. I hope that you can hear me.
Yes.
Okay, perfect. And I hope I'm sharing the screen now. And if we have everything, I can start.
Yes, please.
Okay, thank you. Thank you for the introduction. Thank you also for the Hugo's presentation, which is a very nice introduction to the things that I'm going to say. And I will tell shortly about the eradication of Triosera maculipennis from our Sosky station and how is it going. And as Hugo already mentioned, it won't be easy thing. Shortly, as you know, the Antarctic environment is defined by being isolated, geographically isolated with very harsh climate conditions, limited ice-free areas and short vegetation season. Therefore, the biodiversity of those ice-free regions is very low, and the ecological structure of communities is simple, with limited competition for resources. And due to the growth of scientific and touristic activity together with the climate change, this region recently has been vulnerable, as this session actually is showing, is becoming vulnerable to non-native species introduced accidentally and not only. And increasingly, non-native species have become a major threat for Antarctic communities. And while most alien species cannot survive in Antarctic conditions, unfortunately, several have been able to adapt, and one of them is Triosera maculipennis. I will skip introduction as it was already done. I will just mention and emphasize that it looks like those steady temperatures oscillating around 0 Celsius degrees seems especially important for this fly during the development of its eggs and larvae. And again, as it was mentioned, this fly started to appear first time in Arctica in 2006, and since then was reported from, I guess, right now, all or most of the stations in Maxwell Bay, but also in 2017, it appeared in Herny-Karsowski station, which is located around 25 kilometers from, let's say, for example, our Arctica station. So far, what already was mentioned, but I will emphasize, the Herny-Karsowski station is the only station in Admiralty Bay where Triosera is reported. And aims of my presentation is to first share information about this fly presence in our Sosky station, and methods that we incorporated to monitor and to eradicate it, outcomes of those efforts, and some conclusions are further plans. So shortly introduced, the first record was reported in 2017 in late October in sewage main sewage box. It is located here, the orange star, close to the main building, which is operating all year round. And so far, flies weren't reported never in other sewage tanks that are separated from the main one. And this first report, the flies, the species was confirmed again, as Hugo mentioned, not only on morphological, but also on basis of molecular typing by Marta Portocka and Klausers. And during this first record, the temperature in the tank where flies were found was measured, and it was slightly above 0 Celsius degrees, while the temperature outside was several degrees below 0. At that time, the individuals that were found were both adults that were also found around the sewage box, sewage opening, and larvae that stayed inside. As you can see on the photo, they stay on the wet wooden surface inside the cover box. And at first, regular reports started to appear since 2018. But as you can see, the number of individuals that was found at the beginning was really high and unnerving. And both larvae and imago individuals were found at the time. And with time, after incorporation of eradication efforts, we don't see any larvae, we cannot find any larvae in our Sosky station since 2018. However, adults are still found in very small numbers. Usually it's like 1 up to several individuals per month, but still we find them usually in summer months. And we find them in different places. The first initial finding in septic tank was also for the first months, it was the highest, both larvae and adult individuals. With time, after treatment of the septic tank, we don't find any larvae and any flies there since the beginning of 2020, which is great. However, we still find flies mostly in food storage in main building. And again, as you can see on the other graph, in main building, we find them mostly in food storage containers. And single individuals were also found in kitchen, common room, close to the entrance, in private room and laundry, where they probably spread through the ventilation ducts. There were single individuals found in a small building, meteorological observatory that is close to main building, and also outside of the outside of buildings. What is quite unnerving is the fact that we also twice flies were found in Antarctic specially protected area close to Artsovsky station, in the distance of more or less 1 up to 2 kilometers from the station. So far there were 2 events like that, and but that might be the indication of what Hugo mentioned, so that there is a risk that flies are able to breed at this point outside of human settlements. And hopefully those 2 events were only dispersion, little spontaneous dispersion from station. Measures that we take in Artsovsky station to eradicate those flies are in accordance with those mentioned actually with the paper that Hugo mentioned. And we follow recommendation for education, monitoring, reporting, and we also try to reduce the dispersal of flies. In terms of education, we educate both station personnel and all the guests that are visiting Artsovsky station through lectures before going to Antarctica, while being there, and also both employees and visitors are exposed to posters that are in station, and also to leaflets that we share with everyone who's there. We monitor the presence of the flies using sticky traps all around the station and in all of the buildings and around, and also through obviously personal observations. If someone sees flies, reports it and also tries to catch them and get rid of them. And what we don't do from the recommended things is ultraviolet traps, which we've tried, but unfortunately with conditions in Artsovsky at this moment, because of the safety of the personnel, we resign from that. And also we don't do constant temperature monitoring in the water treatment chambers. It was happening several times, but since there are no flies there, that this thing was stopped and it's not we don't continue it. Station employees report every single situation where flies are found to the Department of Antarctic Biology, and yearly we do report a summary of those findings to Antarctic Treaty consultative meetings and to CEP meetings through information papers. And to reduce the dispersal of flies, sewage tank is regularly emptied and cleaned, and it looks like this really helps. We don't find, it looks like that was the main breeding area for flies, and it's not anymore. However, the problem is we can't find the main source of larvae at the second. And the answer for that might be a new sewage system that will be built due to the ongoing renovation of the Artsovsky station. So fingers crossed. And also another thing that will happen is grids that will be placed in ventilation ducts, hopefully to stop or at least slow down the dispersion of flies. And in conclusion, despite the efforts of the staff at Artsovsky station, so far the population of Triosera remains at a low, very low, however, unfortunately, constant level. And localization and elimination of the breeding places is absolutely necessary and still needs to be done. There is a risk that this fly's life cycle is completed in a natural environment, and this is a thing that has to be researched and checked, and further action to eradicate the fly is necessary and will be carried out at Arsovsky Station. So thank you for your attention, and if you have any questions, I'm willing to answer them.
Thank you very much. I don't see a question in the chat there, but I have a question, being a marine biologist. What is the actual, like all invasive species are bad, but what's the actual threat of these becoming established? What's the danger?
The huge risk in if the Hugo's research, maybe Hugo is actually the person to answer that, but if it looks like this fly is actually adapting to live and establish its population in Antarctica, not only in surroundings where in the stations, but also in the environment, it might potentially took over and changes the balance between the species that might be threat for the species that are already there. As you know, it's the main problem with invasive species, that sometimes they are invasive because they adapt faster and better than the native animals, native species.
Thank you very much. Well, I think with that, that brings our session to a close. I'd like to thank everyone who talked. I think everyone will agree that it was a very nice, varied and interesting session. I'd really like to thank the IT staff. Thank you very much for keeping us running smoothly, especially me not being able to log on at the beginning. Again, I would encourage anyone to go over to the posters and see. We have several posters connected with this session over there, so go over and have a look. This talk will be available on the YouTube channel, I think, in a week or so. So if you've missed anything or want to go back. So yes, so just thank you for everyone for joining and coming to our session. Thank you very much.
Thank you very much.
Bye.
Thank you.
Thank you very much.
Bye, Jamie. Bye, Chiara.
Bye-bye.
Ciao, ciao.
Thank you very much.
Ciao.
Bye, Chiara.
Thank you. Thank you to Jamie.
Thank you, Jamie.
Thank you. Thank you.
Bye. Thank you.
Great job, guys.
Thank you.